Hi, I'm Larry Johnson. I am a professor here at Texas A&M University, and today we're going to talk about the liver, gallbladder, salivary glands, and pancreas. Specifically, we'll be talking about the exocrine and endocrine functions of the liver. We'll be talking about the portal radicles, central vein, blood flow, bile flow, liver cell structures, ultrastructure, and the space of these.
So the liver is the largest gland, and you can see the hepatocytes in through there where the sinuses are going down in between there. You can see the holes in the sinuses, and there then it goes into the central vein. So it produces bile salts. Also, the endocrine function, it produces albumin. Albumin is very important for osmotic pressure of the blood, and also fibrinogen, clotting factors, things like that are produced directly in the blood as endocrine. So here we can see the makeup of part of a classical lobule with the central vein in the middle. And this is your triad. There's three thought parts to it. You got the branch of the hepatic artery bringing oxygenated blood that mixes with the portal blood that's unoxygenated because it already come to the small intestine, and then that percolates through here, and nutrients are then stored in the hepatocytes, and also toxins are detoxified in this process. So the blood goes from these portal radicles to the central vein. However, the bile is in the bile canalicus, and that goes back toward the portal radicle, opposite from the direction of the blood, and it is in a bile duct for it to be discharged in the stored in the gallbladder and then discharged in the small intestines. And here we can see the portal radicle, the bile duct. You can see the cuboidal cells in through there making up that duct, artery, and the hepatic vein. And you can see cords of cells in through these are hepatocytes. This is the blood sinuses that are on either side of those cells. You can see there are individual cells lined up, touching sinuses on either side, and also the bile canalicus is in between to two different cells. Here we see the classical lobule again, with connective tissue surrounding, separating those, and you can see the three components of the portal radical, which is located here and here and here, all around, and all these contribute to blood in the central vein. So if you see one of those as portal radical, you can see the bile duct, hepatic vein, hepatic artery here, and that's the same as here. And then blood would be percolating down through here, ultimately going to the central vein that we're seeing right here. And these are the hepatocytes that we see. Another specimen, we can see the same thing, the bile duct, the artery, the hepatic vein, which is a part of the portal system, hepatic portal system. You can see the cords of cells located here and the central vein. If we look at the different cells that are there, the main cell, the main parenchymal cell, is the hepatocyte. You can see its nucleus, and there's the limits of the cell with lots of mitochondria in there that we can see as the dots that we can see there in this electron micrograph. You can see sinuses in through there, and also you can see these little spots, little white spots here. That's the bile canalicus. And so we can see hepatocytes in different places. Also, we see Kupffer cells here. They have taken up carbon, so they're phagocytic cells that line the sinuses too. You can see one here, usually across the sinuses, as you see there. And then we can see where they've picked up debris in this electron micrographs. So these are cells that are derived from blood monocytes, and they are macrophage-like cells, which are part of the lining of these blood vessels. And then you have endothelial cells. Those endothelial cells are the additional cells that line the sinuses that are there. You have Kupffer cells and the endothelial cells lining the sinuses. And here you can see them all together. The endothelial cells, the Kupffer cells, and the hepatocyte. If we look at the hepatocyte here, we can see big mitochondria through there. Also, see a bile canaliculus, which has tight junctions on either side to keep the bile separated from the rest of from the blood. And here we see lots of glycogen stored in the liver, lots of rough endoplasmic reticulum for making proteins, also some Golgi apparatuses. Here we see a gap junction between adjacent cells that we can see lipid that's there. Also, there's a fair amount of smooth endoplasmic reticulum. I can't see it right now. Nucleus up here. And the smooth endoplasmic reticulum is associated with detoxification. Here we can see one of those sinuses in through there, the hepatic sinus, and the endothelial cells that line that lumen. Just inside, we have the space of Dease. This is a space of Dease here, where microvilli project up from the hepatocyte into the fluid that comes in through. This is sinusoidal type, so you got gaping holes between the within the endothelial cells that allow fluid to bathe the hepatocyte, but it's kept separate from the contents of the bile canaliculus. And here we can see this is the hepatic sinus right into there. There's a platelets going through, and then there's a space of Dease, and these are the endothelial cells, and this is the bile canaliculus and hepatocytes, as we could see.
We want to acknowledge the original source of figures, images, and drawings that might have been used during this presentation. This is the source that they have been identified on the individual slides. This is the end of the liver, gallbladder, salivary gland, pancreas, endocrine, exocrine functions, portal radicles, central vein, blood, bile flow, liver cell structure, ultrastructure, and the space of Dease.